Hi everyone, welcome to our today's webinar. We are honored to have Dr. Stefanie Kickinger with us today. Stefanie, thank you very much for accepting my invitation. First, please allow me to introduce you to our audience. Dr. Stefanie Kickinger did a PhD at the University of Vienna in the Pharmaceinformatics research group of Professor Gerhard Ecker. She finished her PhD last year and received a Lundbeck postdoc grant. Now she's doing postdoc research at the University of Copenhagen. Together with Professor Petrine Wellendorph, she's developing two compounds to inhibit or modulate nutrient amino acid transporters. Today, she's going to talk about transporters as drug targets and how computational methods guide compound design. We will be running a Q&A session at the end of the webinar. If you have any questions in the meantime, please send them into the chat box. So Stefanie, thank you for being here, and without any delay, now the stage is yours.
Okay, thank you so much for this very kind introduction. I'm really excited to be here and to share, my research today. And as you already said, everything will be today about transporters as drug targets, and I'm going to share some, recent examples of our research that we just recently published. So let me start by giving you a little introduction to transporters. So in the human genome, we have a really huge transporter family. It's called the solute carriers, or SLC transporters. And this family is actually the second biggest gene family in the human genome. We have more than 65 families that have more than 420 members. So when you look at the human genome, only the group of the GPCRs have more representatives. So this is like a huge gene family. And in general, transporters are really interesting drug targets because they are expressed, most of them, at the plasma membrane. So they're very important for cell-cell communication, which makes them intrinsically very, important drug targets. And I already said we have about 450 members of these so-called solute carrier transporters, and their major function is to facilitate the uptake or the export of nutrients, ions, metabolites, and drugs. Because they're also, transport drugs, they're also very relevant for so-called drug-drug interaction. So in my talk, so I wanted to also share with you, because the thing is about the transporters is, even though we have more than 420 different members, within the evolution, we only have a very limited numbers of different folds these transporters adopt. So the thing is that the evolution, she was much more, diverse in designing different sequence identities. But when it comes to the fold and the function of the transporters, we are actually very limited. So there's just a few folds known. And so with the folds also comes different, functions. And there are particularly 3 different folds and equally 3 different methods of transport that are very relevant that I wanted to mention, because if you know these 3 different types, you're already, very well equipped to talk to people from the transporter community. So the most important and the most known fold with the most representatives is the so-called MFS fold, so major facilitator fold. And this fold is the first fold here. So I already said, forgot to say that there's about 6 different folds known, and 3 of them are like the most major folds. And I said the MFS fold, so this contains of 12 transmembrane helices that are, we have some kind of pseudosymmetry here. So we have 6 helices that can be superimposed to other 6 helices. And these helices perform like a major conformational fold where they can transport a substrate through the transmembrane. And for example, some interesting representatives are the SLC2 family or the SLC15 family. Then the second fold, also the fold that I was most interested in my PhD and also my postdoc work, is the so-called LUT fold, named after a bacterial homologue that was already crystallized in the early 2000s. And this is also so here what's interesting about that fold, apart from major conformational change, we also have so-called gating residues that help, the substrate pass through the membrane. And I mentioned here we have, for example, the SLC6 transporter family, which is also the family I'm going to focus on today, because this is by far probably the most well investigated transporter family. But here we also have the SLC7 family, which is, for example, a large amino acid transporter, LAT1, which is a really important cancer target. And lastly, a fold that I also want to mention is the so-called elevator fold. And I think that's the most interesting fold, because here the transportation of the substrate really functions as like an elevator motion. So I think it's really also fascinating, how creative nature was in designing these different transporters. And I'm mentioning the different folds because since we know that there is a limited number of folds, we can also, even though we don't know the structure of a certain transporter, but we know they are related to a certain family which share this kind of fold, we can also, retrieve information from this. Okay, and this brings me back to the overview of the different SLC6, SLC transporters. And here I want to particularly focus on the SLC6 family, which is probably the most well investigated family. And when we have a closer look at the 19 different transporters that are within the SLC6 family, we see that we have 4 different subgroups. And let's start with the monoamine transporters, so the transporters for dopamine, serotonin, norepinephrine. So these are really important drug targets, particularly I would say the serotonin transporters, which is the target of interest for all the antidepressants, so the serotonin reuptake inhibitors. We also have now for a few years already a human structure, so we have a very good understanding how this transporter looks like. Then we have the transporters for the neurotransmitter amino acids. So here recently, there was a little breakthrough. So last year, we also now have the structure of those transporters. This is the structure here of the glycine transporter 1. Glycine transporter 1 is particularly a very interesting target for treating schizophrenia. And then we have here in pink, the family of the nutrient amino acid transporters. So this is actually also the family that I'm going to work on now in my postdoc. And here, quite interesting, because of the COVID crisis, we now also have a structure for SLC6A19. So this is a nutrient amino acid transporter. And the reason for that is because apparently this transporter is co-expressed with ACE2, which is the entrance protein, the target of the spike protein of coronavirus. So it's really exciting how different fields of research and actually, happening, influencing each other. And I was really excited to see that because of the Corona crisis, we actually also got an additional transporter structure. And this also inspired me back then to write my grant about this whole family where we're going to, investigate and finding new inhibitors. And then lastly, we have the family of the GABA transporters. And this will also be the transporter I am going to focus this today, because these were the transporters I've been working on the last 5 years. And we have 4 different GABA transporters. Also very interesting that the evolution designed 4 different substrates to basically, transport the same substrate. And then next to the 4 GABA transporters, we also have the transporters for creatine and taurine. And then I also want to give you like a little idea about the sequence identity, so the similarity of these different transporters. So in this graph, you can see the sequence identity of the different subfamily color-coded. And at first sight, you can see that actually the GABA transporter subfamily, so that there's a most color is like bluish and whitish. So this is actually the family that is most related to each other. So from this, you can already conclude that designing selective inhibitors for that kind of family is particularly difficult because they share such high sequence identity. And also a little, interesting fact is that the GABA transporter family is actually the last family of the SLC6 family that does not have a structure yet available. And this is actually, very sad, right? But the reason is probably because they are particularly, difficult to crystallize. So now a little bit about, the relevance of the GABA transporter. So why did I spend, the last 5 years of my life investigating those 4 transporters? So the GABA transporters are expressed, all of them in the brain, but the GABA transporter 2, GAT2, what we also call it, and then the transporter BGT1. So BGT1 stands for beta-in GABA transporter, so that one can also transport beta-in next to GABA. These two are also expressed to a high extent in the liver and the kidney. But in my work, I was so far really interested in their impact, in neuroscience, so in the brain. And when we want to, look for the GABA transporter, we have to actually look to the synapse, so to the neurons and the synapse. So the synapse is particularly where, two neurons talk to each other. So we have the presynaptic neuron and the postsynaptic neuron. And the GABA transporters are particularly expressed in the presynaptic neurons. And once we have an action potential, so there is a signal, then normally our neurotransmitters, which is also GABA is a neurotransmitter, they are released from these vesicles into the neuronal, into the synaptic cleft. And now the major function of the GABA transporters is to facilitate the uptake from GABA from the neuron, from the synaptic cleft back into the neuronal cells. And as we know, GABA is the major inhibitory neurotransmitter in the brain. So GABA normally function as a agonist to so-called GABA-A receptors. So these GABA-A receptors, they are sitting on the postsynaptic membrane. So normally GABA is, released into the synaptic cleft, and then it will here interact with these GABA-A receptors, and then these GABA-A receptors will, they will transport, there are like ion channels, and they will transport chlorine ions into the postsynaptic membrane, and this will result in an increased, in a decreased membrane potential, making the postsynaptic neuron less sensitive for signaling, right? So this is why we have an inhibitory effect of GABA. So the thing is, once we inhibit the uptake, the reuptake from GABA from the synaptic cleft back into the neuronal cells, we can increase the levels of GABA in the synaptic cleft and therefore increase its inhibitory effect, because we have now more GABA that can all interact with these GABA-A receptors, right? And this pharmacological concept has been already successfully applied to treating certain diseases, for example, epilepsy. So in epilepsy, we have, too much firing of the neurons in the brain. So by increasing the levels of GABA, we can actually, decrease this neuronal firing. And there has already been one drug approved. It's called tiagabine. It's a GABA transporter 1 inhibitor. And it also very much reflects the knowledge that we have today. So we know quite a lot about GABA transporter 1, also because we have this very potent and also already approved drug. But we know quite little about the other GABA transporters. And the thing is, with that drug tiagabine, it has already been developed in the 90s, and it actually has been here developed in Denmark, where I'm right now. But the clinicians do not like to use that drug because it really has some severe side effects, such as dizziness and sleepiness. So my new supervisor, Professor Petrine Wellendorph, sees things that maybe the other GABA transporters are probably, or maybe some better drug targets in terms of treating epilepsy that show less side effects. And particularly, we think that BGT1, so the beta-in GABA transporter, which is located at extrasynaptic sites, so here in astrocytes, close to these extrasynaptic GABA-A receptors. And we believe that this is a particularly interesting target, because once we inhibit GABA uptake here, we can increase the GABA levels around these extrasynaptic GABA-A receptors, and this can then result in what we call tonic inhibition, so a permanent inhibition, which could be very helpful for treating epilepsy. But the thing is, as we don't have a structure and we don't have yet selective compounds, so my PhD, we tried to understand what makes a compound a selective inhibitor, particularly for the BGT1 transporter. So because this is going to be some structure-based work, I also have to walk you through the different structure that we have available. So we don't have a 3D structure, but we have the related structures from the other SLC6 transporters, and we know that they all share the same overall code, because apparently evolution wasn't so creative in designing these different folds. And the fold that we are talking about is these 12 transmembrane helices, and you can take the first 5 helices and you can actually superimpose them on the next 5 helixes. So we have some kind of pseudo-symmetry in our transporter. And I already said that in the beginning of the 2000s, we had the first structure revealed. It was the bacterial homologue, the so-called leucine transporter. And then in 2013, the next structure was released. It was the dopamine transporter of the Drosophila. And then in 2016, right in the beginning of my PhD, the human serotonin structure was released. And that was like a huge breakthrough in the field of transporter in science, because what was also super interesting about that new structure of the human serotonin transporter was that it was co-crystallized with 2 ligands. So one ligand in what we see called the orthosteric pocket, so there where also the endogenous substrate binds, and also a second pocket, an allosteric pocket. So we now also have a ligand here in this allosteric pocket, which is placed right above the orthosteric side. And because the serotonin transporter shares so much high sequence identity with the GABA transporters, we believe that there is also such a allosteric binding pocket in the GABA transporters. And also in my research, we've been trying to prove that there is this binding site. And this brings me also to some very special features of the GABA transporters. So what's really, really interesting about the GABA transporters when you compare it with the other SLC6 transporters is that when you have a look at TM10, so the transmembrane region 10, so here it's colored in orange, is that here, right in the middle of this trans helix, we have a so-called insertion. So all the GABA transporters, including taurine and creatine transporters, we have 1 amino acid more in this transmembrane here. So we have this serine here introduced right in the middle. And this serine probably results in a more extended helix. And the thing is that since we don't have a crystal structure, we actually don't know how particularly, like what is the 3D structure of that insertion, particularly in that area. And what's also super interesting is that that insertion happens to be particularly between the side of the orthosteric and the allosteric side. So we are pretty much convinced that nature put this insertion on purpose there, because apparently it is very, very relevant for selectivity. But when we try to predict, the 3D structures of our GABA transporters, we have to acknowledge that particularly in that area, which is unfortunately very significant because it's in the interface of these binding sites, our models are probably very inaccurate. Okay, and that leaves me one of my last introductory slides, because we are talking transporters. So I also have to walk you through that transporters are highly flexible proteins. And in order to facilitate the uptake, the transport of a substrate, they have to undergo specific conformational changes. And that from a computational perspective is really fascinating and exciting, but also very difficult to predict, right? So a very, the transport cycle roughly contains of 4 different conformational changes. So in the beginning, we have an equilibrium of our transporters that adopt a conformation that is so-called outward open, so open to the extracellular side. So here our substrate GABA, and then the 2 ions, sodium ions and 1 chlorines, which are driving the transporters, they can bind. Once the substrate binds, this triggers the transporter to adopt the so-called outward occluded conformation. So we have this in blue here. This schematic here represents what we call so-called gating residues. So we have really 4 residues that kind of block the compound from the extracellular side. And then we have some major conformational change going on where the transporter adopts the so-called inward occluded conformation. And on the inward side, we also have, again, these gating residues. And these gates can then swing open, and then we have, the substrate release into the inward side. And why I'm mentioning all these different conformations, first, because it's a very important conceptual thing that we understand that a transporter is always only accessible from one side of the transporter. So there's never ever like a channel that we see, for example, in the ion channel. So there's always, the transport is always occluded at least from one side. And second is because from a computational perspective, we have to rationalize, okay, what is the actual transporter conformation that we are interested in at the moment? And I already said that in my PhD, I was interested in designing inhibitors for the GABA transporters and to understand how they are selective. And this is why I was particularly interested in the outward open conformation, because this is why we believe inhibitors, particularly bulky compounds, they bind to this outward open conformation and they kind of trap the transporter in that conformation. And then we're also interested in the outward occluded conformation, because we believe that this conformation is the state where a so-called substrate binds to compound that are actually transported. So we believe that at some point they have to fit in this occluded conformation. And then I just thought I'd briefly mention the different computational methods that I've been using in our compound design. And so I've been doing a lot of what we call homology modeling. So homology modeling is when you take the backbone of a known substrate that shares the same overall fold with your protein, and you basically model the sequence identity of your desired protein to this. And here I also have to mention that there was a breakthrough happening in the last year, and probably many of you are aware, because actually Google has released an algorithm that basically flipped everything around. So now Google can really, really well predict the 3D conformation of our proteins just by inserting the sequence identity. But for a very long time before Google was there, and in my PhD, we did it like the old-fashioned way, where we did homology modeling, where we took the known structure of a known compound, and then based on the structure we were modeling it. And I will also talk about Google Fold in a minute, because it was so groundbreaking, right? But here I just want to give a little introduction to the old thing, how we did it before Google AlphaFold. And here I still want to mention again, I was already talking about that insertion in TM10, so in the transmembrane region 10. And here we know that our models are particularly inaccurate because we do not know how this insertion works there. And I'm also pretty sure that AlphaFold doesn't know how it works, but I will come to this. And second, I did a lot of molecular docking. So docking is a method that really gives you very fast results in terms of how your compound potentially binds. So it's a very nice tool for creating binding hypotheses. So what you basically do first, you need to define the pocket of your where you believe that your substrate binds or your compounds bind. So this is already a very crucial, decision that you have to make. In the field of transporters, we mostly know where our substrates and compounds bind. So that's actually a very easy step for us to define, the binding pocket. And then second, the algorithms, we have so-called placements algorithms. So they take the different conformation of our compounds and they fit it into the defined pocket. And then the third, and I would say the most crucial step, we have so-called scoring algorithms that basically assign or basically score and assess the fit of your compound to the protein. And here they basically, what they do, these algorithms try to assess binding free energy. And they do that by simply checking, okay, how many interactions do you have? Are there any clashes and so on? And then they sum up basically the energies. And it's a very brute force method, but it's very fast. And it's a method to gain like a lot of interesting ideas. Right? But the thing is that with docking, you always have to know, okay, the problem is that any algorithm probably can always fit any compound anywhere. So you always have to be a little bit, you have to always question whether what the algorithm does actually makes sense for you. And then lastly, what I've been also doing a lot is that we take what we call, the pose of a compound. So this is like what we believe is the most accurate representation of our binding method. So we take these poses and then we perform so-called molecular dynamic simulations. So in molecular dynamics, we assume that all our atoms are balls, and then we can apply, the motions, Newton laws of motion, and we can predict the movements of our atoms. And we do that for very small time scales. So for now, if you are going, so what I did in my PhD, I simulated for 100, 200 nanoseconds. There are now groups out there that can actually, also simulate for microseconds and some even milliseconds. And when it comes to the transport event, we have to make ourselves clear that this is actually quite slow events, right? So a transport will probably take a few milliseconds. So this is why we're still having issues simulating the whole process. But in the work that I was doing, we were not interested in the overall motions, but we were interested in seeing whether once we predict the binding mode, whether we can see that the interaction that we observe are also stable over time. So for this kind of research question, it's still okay to only simulate like 100 or 200 nanoseconds. So you don't have to do milliseconds for that. Okay, and this brings me already to the different research angles that we have been taking, particularly regarding the beta-in GABA transporter, so BGT1. And I already said that we think that there are two binding sites in BGT1. So we have the orthosteric binding site, and we've been developing different compounds for that orthosteric site. And there's this postulated allosteric site where we have also developed as non-competitive inhibitors, so where we believe that they bind somewhere else than the orthosteric site. But for the talk, since I only have limited time, I decided I'm going to share our newest publication, which is where we identified the binding mode of this compound, which we now call bicyclic GABA. So this compound is so far most potent and most selective inhibitor that we know. And this was a huge collaboration between my former supervisor, Professor Ecker, and also my current supervisor, Professor Wellendorph. So what did we do? I also have to mention our chemistry collaborator. So this compound was actually designed by the students of Professor Satoshi Shuto. He is a professor of chemistry at the University of Hokkaido. And they first developed this small compound here. We call it compound number 1. And this was already a quite good inhibitor of the BGT1, so with 5.6 micromolar. And then they decided to conformationally restrict this small compound, and then they designed bicyclic GABA, and miraculously this compound turned out to be like a really potent inhibitor, so 0.6. Regarding the potency, I also have to say that, if you're, for example, in the field of GPCR research, you would say 0.6 micromolar, that's like not so potent. Well, in the field of transporter research, particularly in GABA research, our potent compounds are probably around the micromolar range. And this one with lower micromolar range, this is already like super potent in our field. So what we did next is I performed docking and molecular dynamic simulation of these two compounds. And then based on the first preliminary binding hypothesis, we designed some mutational experiments in order to prove that, the residues that we believe interact with our compounds are particularly, the residues relevant for binding. And then once we have confirmed this binding hypothesis, we use the binding hypothesis to also design new compounds. Because, since you can imagine these compounds are really small, they are also, double charged. So this carboxyl group is decarboxylated, and this nitrogen is protonated along with human pH of 7.4. So we suspect them to not be really, brain penetrating. And since we are talking about, the synapse here, we really want them to go into the brain at some point. So we wanted these compounds to be, more lipophilic, right? So this is also the point where I think it's, I have to mention and talk about AlphaFold. So as structural biologists and also structure-based design people, it is now, I think, our responsibilities whenever we publish something, we have to consider why we did, our own homology models, or why are we not using the structures of AlphaFold, right? I said that AlphaFold did a really amazing job in predicting the whole human genome. So you can just Google it and go there and check all of the structures in the human genome. And so they also predicted the structures of all the GABA transporters. And I performed, of course, first thing I did, I performed an alignment, and I observed something quite interesting. So when we look at the GABA structures, the GABA transporter structures, we see that they do, like here I have fitted an outward occluded model. And it fits quite nicely, but we can also see that there is some inaccuracy. And when I compared outward open conformation, so the blue one is again always the AlphaFold structures, I could see that they much more nicely align. So what I observed and what I conclude from this, that apparently AlphaFold is biased in predicting the outward open conformation. I've been talking to other colleagues that are working with different folds of transporters, and particularly for the MFS fold, the biggest fold family. There apparently is a bias toward the inward occluded. So whenever you are going to work with the AlphaFold structures, make sure that you are aware of the different conformations of your target, because apparently AlphaFold is not so good yet in predicting these different folds. And the reason for this is probably also because AlphaFold was trained on the structures that are already available in the PDB. And for the transporters, particularly, or the SLC6 transporters, the most available fold that we have in the PDB is still the outward open conformation. And this is probably also why AlphaFold has been predicting this conformation. And then I was already mentioning quite a lot, this transmembrane region 10, where we have this insertion. And what is also interesting that I've been seeing so AlphaFold is doing some quite weird things here. So I'm pretty sure that AlphaFold also doesn't know how to model this insertion. And we have some experimental studies from other groups where they predict that this insertion should be what we call a pi helix, or a more extended conformation. So I'm also pretty sure that AlphaFold is actually poorly predicting this area. So once you investigate AlphaFold structures, be aware that you're knowing where, what is the alignment of your structure to your desired protein, and if there are any insertions or deletions, so where your, for example, your helix is a little bit shorter, right? So from this, you can then also conclude, okay, is this area particularly trustworthy? So in terms of the AlphaFold structures regarding, BGT1, I think it's still valid to use a conventional homology modeling, because you can argue that you can investigate, a little bit deeper with homology modeling how this insertion particularly should look, and also in terms of the different conformations. And in the project I'm going to talk about, like the docking that I did, we were going for the outward occluded conformation. The reason for that is because we had these super small compounds where we are pretty sure that they fit into that occluded state. So for this, it's also, I think, justifiable that you say, okay, since you want a different conformation than the one that AlphaFold predicts, it's okay to still use a conventional homology model. Okay, and this already brings me to our actual binding hypothesis. I said that we did docking. So what we normally do in transporter docking is that we use an induced fit protocol. And the reason for that is because we want to address the intrinsic flexibility of the transporter, and also, the uncertainty we have, particularly in the TM10 area. And when I did the docking, I observed that there was this glutamine 299 that we see some hydrogen bonding, and this glutamate E52. And then, of course, I also have to mention there's this sodium ion in the pocket. So this is a feature that all of the SLC6 transporters share. And we are pretty confident that our carboxy group, which is also a key feature of BGT1 inhibitors, this carboxy group is actually always coordinating in sodium ion. And once you know such a key interaction, it's actually pretty easy to model the rest, because then you already have less options, right? And what was also interesting for the, the precursor compound, the smaller one, we also predict the more or less same interaction with this glutamate. And then also in the simulations, when I simulated these compounds for 100 nanoseconds, I could also confirm that these are interaction, this glutamate and that glutamine, also for the small compound, that these interactions are also stable over time. So in the next step, based on this hypothesis, we also suggested that we can do some, derivatives, and to, guide the medicinal chemists on where to put, some bulky compounds, so we can make these compounds more lipophilic. And then we thought, well, let's go for like a derivative that is placed somewhere outside the bucket. So we know that these are the gating residues, and we know that they are rather flexible. So these can actually move, and we believe if we add some bulk here, that we can actually extend the compound in this so-called extracellular vestibule. So this is where the entry pathway of our compound is. And we know this from other SLC6 transporters where this actually works. So we thought that this would actually be the most promising side of derivatization. But the problem was that this, particularly this carbon atom, was super, super difficult to derivatize. So the chemists couldn't make that compound. So that we had to, approach a different strategy. And the thing is that we observed, well, we have also the nitrogen here, and we believe that we have a little bit of space here in the pocket, and this is where we also suggested to the chemists that they should do some derivatives of the nitrogen here, and this is exactly what they did. And we said that we believe that the space is very limited at that site, so they have to do only very small derivatives. So we added 1 or 2 methyl groups here. And what's also interesting about that nitrogen is that because this is a GABA derivative, right? It's basically a cyclic form of GABA. We also suspect, and we know that this compound also hits GABA A receptors. So we have off-site pharmacology, and we know that by introducing bulk at the amino group, we can actually also decrease the off-site targets. And then for the thing is that we were really interested in probing our hypothesis that we can extend the compound, into that extracellular space. And this is why we went back, to the precursor compound. And we know that this carbon here is actually, placed similarly than this carbon. And we hope that by attaching bulk here, we can actually, fit into that extracellular vestibule. So this compounds we just designed to basically, follow up on our hypothesis that it's possible to add some bulky stuff, that extends into the extracellular vestibule. So this is why we designed some compound with methyl, ethyl, and phenyl at this carbon here. And then we did our initial screening, and yeah, we were pleased to see that actually the compound with the methyl at the nitrogen of our bicycle GABA derivative actually showed some inhibitory activity. So what you see here is the different compounds on the x-axis. We tested them initially at 100 micromolar, and then we have the uptake of GABA. And so the more inhibition we have, the more potent our compound is. And we chose a cutoff of about 50% for selecting, potent inhibitors. And we saw that actually only this compound 2 with the 1-methyl was actually active, whereas the ones with the 2-methyl were inactive. And from this, we conclude apparently our hypothesis was right, that we only have limited space down there in the pocket. And then from our small derivatives, we saw that unfortunately only the compound with the methyl group here showed some activity, and the others with the phenyl and the ethyl, they were both unfortunately dead. So here we concluded apparently we were not able to really predict that these compounds are able to extend into that extracellular vestibule. And this also kind of confirms that we have having apparently some issues also with the accuracy of our models in that extracellular vestibule, which is close to what I already said about the transmembrane region 10. So we decided that we're going to follow up on our 2 compounds that showed some promising activity. So our original compound, bicycle GABA, and the derivative 2, where we have the methyl group of nitrogen. And then we first checked, okay, these 2 amino acids, glutamine and glutamate, okay, are they conserved among the GABA transporters? And we saw that actually this glutamate here is not conserved among the GABA. It corresponds to GET1. It corresponds to tyrosine and GET1, and that glutamine is particularly interesting because that's a unique residue for BGT1. So this glutamine corresponds to leucine and all the other transporters. And this is why it was very straightforward for us to design some mutants. So first we said, well, let's design this glutamine to leucine mutant, and we would predict to, decrease activity for a compound because we're going to, kill this one of these interactions here, this hydrogen bonding. And then we also said, well, this should also work the other way around. So when we take GAP3, where our compound has really low activity, and introduce that glutamine at the same position, we should be actually able to introduce activity. And then for the glutamate, we said, well, since it's a tyrosine in GAP1, we will, turn it into a tyrosine, and also again predict to decrease activity. And then on the other hand, we said, let's do a more classic exchange and also do it into alanine and also predict to decrease. And lastly, we also said, let's go for a double mutant where we would also, predict to decrease activity. And when the experimental results came in, we were actually very pleased to see that, we were able to, decrease the activity of our compound for the single mutant. So when we look at pharmacological so-called dose response curves, so let's see here, we have here the different concentrations of our compounds. So here of bicycle GABA. And here we have the uptake, so 100% uptake means there's a lot of uptake of GABA, and then low concentration means low uptake of GABA. And what we would expect here pharmacologically, if we decrease activity, we should see a right shift of the curve. And this is exactly what we saw for all the single mutants. So we saw a decrease in activity. So here we have the IC50 values, and also for the double mutant. Interestingly and very strikingly, for this mutant where we turned the glutamate into alanine, what we saw was a ridiculous increase in activity. So this is really something really fascinating, and up to this point we are not actually able to explain what is happening here. So for some weird reason, if we turn that glutamate into alanine, we see a ridiculous increase in activity. So very fascinating. And for bicycle GABA, this worked out pretty well, and for our derivative 2, we actually saw a similar trend then as for bicycle GABA. And then when we look at the GAT3 mutant, where we introduced that glutamine, we were really happy to see that we could drastically increase our activity. So what we see pharmacologically here, we should see a left shift of the curve because it's getting more potent. And because we were able to introduce activity in GAT3, we believe that this is some really good evidence that glutamine is a key residue for interaction, because it's always more difficult to introduce activity rather than getting rid of it. And unfortunately, when we look at our derivative 2, we were not able to introduce activity, right? And we believe the reason for that is because derivative 2 is less selective than bicycle GABA, and we believe that probably this is related to the fact that once we have a methyl group attached here, we can do fewer hydrogen bonds, and we believe that's the reason why it's less selective and why we weren't able to really introduce activity here in GAT3. So this already brings me to the conclusion. So as I said, we already found that glutamine particularly, and also that glutamate 52 are relevant for activity and selectivity. So we now believe, and we know, that there are key features for BGT1 inhibition. Then something that I haven't shared with you yet, but which is pretty exciting, is that bicycle GABA is not transported by BGT1. So our collaborators, they performed some so-called FMP assays, and they could see that bicycle GABA is not transported inside the cell. And this is so remarkable because that compound is so small and it looks exactly like, it's just a GABA derivative. But knowing that it's not transported, this gives us, no more options. So what we did, and what we are right now working on, is that we actually got a radio-labeled version of bicycle GABA. And this is super exciting because right now what we're doing are actually binding experiments. So I hope that in the soon future, I can also share these results with you people. And then lastly, what we also have to acknowledge, and this is also always really painful, when you have to acknowledge that something did not work out. So what we saw is that our bulky derivatives, that we failed actually to extend these compounds into the extracellular side. So our predictions were all wrong for these kind of compounds. And we have to particularly acknowledge that this is because our models are very inaccurate in that certain area of the transporter, and this is probably related to that insertion in our compound, in our transporter. And lastly, I already mentioned that we have this really fascinating increase in activity of our compounds for this glutamate to alanine mutant. And even though we cannot explain it, we really believe that this is a fantastic candidate for cryo-EM or X-ray studies, because by drastically increasing the activity of our compound, we think that it's probably easier to get, a protein ligand complex. So we already have a collaboration, and this is already ongoing. So hopefully soon there will actually be a structure. And once we have a structure, we hopefully also know that at some point, we can, we know how the 3D structure of that insertion is. And this leaves me then only to acknowledge the two fantastic groups that I've been working the last 5 years with. So my former supervisor, this is the Pharmaceinformatics research group. You see, it's a really huge group. This is, my former supervisor, Gerhard. And then I also have to, say thank you to my new group. So this is the group of Petrine Wellendorph. This is Petrine, and you can see they are very female. There are only a few men that on that day particularly were not there. And yeah, lastly, I can only say thank you for your attention. And then I also want to share with you that we are having, we are organizing a conference. So the International Transmembrane Transporter Conference, it was supposed to already happen 2 years ago, but we are pretty confident that it will actually happen next year in Copenhagen. And I also put a picture of Copenhagen in the back. So Copenhagen is a lovely city. So it's going to happen next year from June 7th to 10th. And I hope that I might see some of your people there. So thank you.
All right. So thank you very much for the wonderful talk. And I think everyone now has far more understanding about the topics. And now we have some questions in the chat box. So the first one is how to predict the structure of dimer.
Oh, that's a good question. So with the GABA transporters, we know particularly GABA1 is also functional as a dimer. And there are some studies out there not related to GABA transporter 1, but to the bacterial homolog, the LUT, and also to the serotonin transporter. And I think that we probably have the same dimerization sites also in the GABA transporter 1. But in my research, it was not that relevant because we know that the functional unit is actually the monomer. So for predicting binding modes and inhibitors and so on, we do not need the dimer.
Yeah, okay, thank you. And the next one is, some of the audience, they are interested in the basic research protocol on probing molecules in your lab.
So the basic so what we normally do is that depends on if you're working with a few numbers like of compounds where you have, for example, congeneric series. So in that case that I just presented, we had a small number of compounds that shared the same overall scaffold. So what we normally do with these compounds, we perform docking, and then we have a quite elaborate protocol of how we do in clustering, because we assume that these compounds, since they have the same scaffold, that they bind in a similar way. So we normally like to do a clustering of the different ways that compound can fit into the into the protein, and then we look at the different clusters, and then we look at the best scored poses of different clusters. And I think that's a very good protocol to work with when you have congeneric series. When you're doing virtual screening whatsoever, then I guess it's more difficult, but there we also like to do clustering approaches. So for example, in the Schrödinger suite, you can do what we call a volume overlap clustering. So this gives you a pretty interesting idea of, what is the different hotspots in your pocket. And from this, I think it's also a good idea to look at the different different sites where you get really good scores for your compounds, and you can then also see if there's certain amino acids that maybe pop up in different clusters, and then you go into the literature and check if there's maybe some mutational data, and that always also helps you to prioritize. What I really want to stress is that if you're doing particularly virtual screening, and also if you're working with small compound sets, it's not enough to only focus on scoring. So scoring can be really misleading, and what we have seen is that the scoring functions, most of them are also not trained on transporters. So they're mostly optimized for kinases and so on, right? So which are soluble proteins and very different to transporters. So what I can share is, in my experience, that the scoring function really perform horribly for the most transporters. So I hope that answered the question.
Yeah, thank you. Okay, and now we have a question on slide 19. So is it, we can first go back to 19. Yeah, is it possible that the bicyclo GABA alternatively bind to the transporter via an alternative binding mode, whereby the positively charged nitrogen interact with the aromatic cage, like the tyrosine or phenylalanine residues, and could explain the mutation?
So we know that bicyclo GABA is a competitive inhibitor, so it's able to displace GABA from the binding site, and this is why we believe that bicyclo GABA has to bind to that orthosteric site. And we believe maybe it's also related to space, maybe in the pocket, because by exchanging glutamate to alanine, we have much more space there. But it just doesn't make sense at the end of the day, because we are actually killing a major interaction and we see this huge increase. So it's yeah, I just have to be honest here that we cannot really explain it, but we believe it has to be in that site. So we are very confident about that.
Okay, yeah, thank you. And then the next question is, I wonder how to find the binding pocket of a transporter. Is it always a tunnel?
So that's something, so it's never like a tunnel, because when we work with transporters, we have to always to remember the transporters are always only accessible from one side, right? And if you know the fold, and if you know that the compounds that you're working with, for example, are competitive inhibitors, then you can be fairly certain that it is the orthosteric binding site. And for example, for the SLC6 transporters, and I would say for all the Lutey fold transporters, it's very easy to detect the binding site. But there has been more and more research also on allosteric inhibitors, and I also did a study on another non-competitive inhibitor, so compounds that are actually not able to displace GABA from its orthosteric site. And here we are not actually sure. So we know that there's probably this allosteric site that we have already observed in serotonin transporter, but there could be possibly other sites, and there has been also increased sites, in the membrane, like lipophilic binding sites, because they're for the serotonin and dopamine transporters. There are also out there some, fatty acids that kind of, modulate the transport. So there is still a lot to be learned about the different binding sites here.
Yeah, thank you. So our next question will be, how is the docking of small molecules onto the protein helped by AlphaFold other than providing the target model?
So in my study, we didn't use the AlphaFold models, right? And the reason for that is because I think that since we're working with these small compounds that were so fit to the occluded transporter conformation, I think that the AlphaFold models were not the right ones to go here. So whenever you work with the AlphaFold models, and I think you should, I'm not saying you should not, right? So in my new project where I'm working on the nutrient amino acid transport, I'm going to use the AlphaFold models, right? So you always have to make an argument, particularly if you're not going to use it. And one reason for that could be the different conformations that you're interested in. And this is always relevant when it comes to transporters. When it comes to other enzymes, for example, that maybe only exist in one kind of conformation, I guess there's no point in actually spending time on doing homology modeling. So always be wise in where you want to spend your time.
Mm-hmm. Yeah, thanks. So there's a follow-up question from the same friend asking, so maybe can you give more insight on how AlphaFold develops on the protein ligand field? So is it really complementary to the current methods?
So I cannot really tell you, how specifically AlphaFold does it. So they're using apparently some deep learning methods, and that's not my field of expertise. But what I can tell you is that they're doing a fantastic job. So if you, for example, go and compare the serotonin structure to the AlphaFold model, it's impeccable, right? So they are definitely onto something, and it's going to bring the field. It will, it will fast forward the field, particularly for proteins where we are not yet aware of the folds. So it will be, for example, what I haven't done yet, but I am planning to do is actually looking at all the SLC transporters where we are not yet aware of the fold, and just check what AlphaFold did here. And I think that within the next month we will also see how accurate AlphaFold is to predict these kind of new folds. So it will be, I'm pretty sure it is already like a revolution happening here, and we are actually sitting front row. So it's like a really exciting time right now, and particularly for protein ligands. So the thing is that AlphaFold is right now only predicting apostructures. We will see in the future if this is going to end, to change. Maybe AlphaFold can do it, but of course you also have to take into account that when you look at the structure and you also want to, then predict binding modes, that of course the pocket changes if you have different shaped of ligands in there. But still, the overall folds, they will not change that much, with different type of ligands. So yeah.
Yeah. Okay. Yeah. Thank you very much. So there is no more questions coming into the chat box. So maybe I think we can close up the Q&A session. And so thank you very much again, Stefanie. And yeah, I really liked your insight about the AlphaFold, because I think as a drug designer, I think you need to be really careful about the structure from AlphaFold, because maybe the overall conformation is really correct and really very nice, but maybe the pocket is not that nice predicted. So as a drug designer, you should really check your pockets, and otherwise maybe a homology modeling should be also, yeah, better solution. And once again, thank you on behalf of everyone here. Thank you for taking time out and being here today. And also thanks to everyone who take part, yeah, who are here today. And I hope you have a nice day and nice evening. Yeah, then let's meet us, yeah, next week. Then bye-bye. Yeah, thank you. Bye.
Thank you. Bye.
Thank you. Bye.